So simple math. This is simple math. So here, I mean, don't even, you could write the words down all you want, but you need to understand it. So here is a sphere, and you have to know what a sphere is to be able to understand this unit. A sphere is like a three-dimensional circle, right? So if I got to move this out of the way. These 8 small spheres have a radius of 1, and they have the same total volume as a larger sphere that has a radius of 2. But the critical part here, the last sentence, they have 4 times the total surface area. And when you're dealing with something that needs to absorb as much oxygen like a lung as possible, we need surface area per volume. Okay. This is basically why cells are small. Cells have to constantly take in oxygen. They have to get rid of waste products. They have to get rid of CO2. They have to bring in glucose. And the way they do it is like a worm, they bring it through their skin, except instead of the skin, we're going to talk about what's called a plasma membrane. Okay. So this is a really important slide because it really makes you understand or helps you understand that surface area to volume ratio problem that we're dealing with. Okay, let's look at it in a little bit more mathematical terms. So here's a big cell. Let's just do the big cell. I'm gonna use my special little pen thing. So here's a big cell, and if we're talking about nutrients, we could talk about oxygen. That's me drawing oxygen. It's pretty good, isn't it, Bingo? And then you're gonna get rid of waste like CO2. Oh, come on, there you go. CO2. Okay, so you're going to get rid of all these products. And the only way you can do it is basically through the skin or the plasma membrane of the sphere. Well, when you drop down in size, it's a lot easier to move something from the middle and move it out. If I'm in the middle here and I'm a very large cell, it's very difficult for me to get all that waste product out on the surface area. And what happens is cells literally die if they get too big because they can't get the waste product out and they can't get the nutrients in an efficient, quick manner. Okay, so this is why cells are small. Okay, so here's your volume of a sphere, 4/3 pi r cubed. Here's the surface area of a sphere, 4 pi r squared. So when you calculate, and you're going to have to do this on the test, to calculate the simple surface area to volume ratio, it's 3 over r. It's 3 over the radius of a circle. To give you an idea what that looks like on a graph, here's our surface area to volume ratio. And obviously, a higher surface area is good. So this is good to have. A low surface area to volume ratio is not as good to have. So you want to have, I don't know why I can't draw a plus sign. Isn't that crazy? Okay, this is good to have, this is bad to have. So if you have a small surface area, it's usually because you have a very large cell. Okay, if you have a high surface area to volume ratio, that's usually because that it's a very small cell. So cells usually don't get any larger than 200 micrometers in size. If they get bigger, they die of their own waste product. So as soon as the cell gets bigger, it literally starts to split or divide in half, which we'll talk about later. Okay. So you need to understand. If I ask you a question, can you see me wink through the glasses? Can you see that? Are, are you able to see that? Alyssa, can you see that? Yeah. All right. So if I tell you why does a cell divide, you would tell me because it needs to split to get smaller so that it increases its surface area to volume ratio. Say it again, because I don't know if Draper heard me. Why does a cell divide? So it can increase its surface area to volume ratio to get rid of waste products. Got it? And you notice on my test, they're not right there questions, guys. So you really have to understand what we're talking about. So the basic concept of the past 3 slides were if a cell is small, it's more efficient because the surface area to volume ratio is increased. That's the big picture. All right, let's keep going. So why is this important? Bingo. You there?
Yeah.
Um, it still cracks me up that you're listening to the Bee Gees, but um, what happens with your lungs is if you kind of and don't do you remember when we stuck our fingers down through our esophagus and our intestines and we grabbed the inside tip and pulled ourselves inside out? Do you guys remember talking about that? Shake your head yes, you remember that. Okay. If now there's two tubes going down from your throat. One of them is the esophagus, which goes to your digestive system. The other one is a trachea, which goes down into your lungs. And there's a little flapper that manages it. If you eat food, it opens one flap and it goes to the esophagus. If you're breathing, it goes to the other flap and you're going to the trachea and the lungs. And that branch that goes from the trachea branches into two and it goes to two lungs. And then it continues to branch smaller and smaller and smaller and smaller until you get to this small little guy, which I have right here. And this would be the end point of something in your lungs. And you have millions of alveoli, millions of them in your lungs, and they're very, very, very small. And what happens is they're small because we want to have as high a surface area as possible per the volume given. So as the oxygen goes into these guys, so take a deep breath, everybody, like an obnoxiously deep breath. Okay, basically what you've just done is you filled all your alveoli with oxygen. So you've increased the concentration of oxygen in one alveoli. And what's going to happen is it's going to move through the cell membranes, and we'll see this in higher detail in a second. But it's going to move through the cell membranes of the cell that's around the alveoli, and it's going to go into your bloodstream, and then it's going to be carried away. So obviously, like whenever you need more oxygen, you breathe more often, and that's increasing the amount of oxygen into your alveoli. If if you just had a lung with a really large cell, you would not be able to breathe enough to basically take care of all the activities you do on a daily basis. It simply wouldn't happen. The fact that you have this very high surface area to volume ratio type of system allows us to do that. At the same time, there's carbon dioxide in your blood, which you can see right here. And the carbon dioxide moves out, goes into your alveoli, and then that moves out as you breathe back out. And we'll talk about that in a second. All right. This should be interesting. We all know someone that smokes or vapes, right? And we don't have to raise our hands or do anything like that. We all know someone that smokes or vapes. Basically, what they're doing, and this is, let me give you a picture of what this is. This whole thing that I'm kind of outlining here, this whole thing is a blood vessel. It's a capillary. Capillary means it's a little thinner wall, and it basically is used to move things back and forth between the bloodstream. Capillary is what would be all over your small intestine to get the glucose. In this case, they're getting oxygen, so they're all over the alveoli. So here is the inside of the alveoli. Okay, so this is, they just call it a surfactant layer. It's just basically a layer that's on the inside of the alveoli. Then you have these really thin cells here, and you don't need to know anything particular here. So this slide is just an information slide. It's not a content for test slide, but these are really thin cells. And what happens is the oxygen moves right through that layer and it goes into the bloodstream and gets picked up by a red blood cell. But there is a particular metal that helps the oxygen bind on the blood cell. Anybody know what that metal is that's in our bodies? Shout it out.
Iron. Iron.
Yep, it's iron, and iron is bound to hemoglobin, and hemoglobin's on the red blood cell, and that's basically what's grabbing onto the oxygen. Now, if you smoke, or the smoke is basically going to make a layer of tar all the way through this whole inside layer of alveoli. Because when you smoke, you take a deep breath, the smoke goes down into your lungs, and then that tar cannot move into the bloodstream. Now, some of the chemicals in the cigarette smoke can, and that's what gives you that little bit of a high feeling or a buzz. But the bad side of this is that you get a layer of tar all the way through there. Now, if you have tar, you're not going to be able to move the oxygen through here. And what's tough about tar is tar you can't get rid of. Tar is going to stay there. So if you smoke, that tar will line this inside lining, and the oxygen is not going to move through. That's why you could tell a smoker a mile away, because they can't do aerobic exercises. They can't handle that kind of stuff because they can't get the oxygen into their body. Then eventually they develop that really bad cough. That cough is their body desperately trying to get rid of that tar, but it won't leave. The tar won't leave. So then people go, well, okay, well then, vaping is okay because it doesn't have the tar in it. The problem with vaping is you superheat plasticized water with chemicals in it, the same type of chemicals that are in cigarette smoke, and you're basically heating up a really high resin plastic. And when you breathe in that high resin plastic, that plastic now is getting stuck inside of kids' lungs, especially kids your age. They're getting stuck inside there. They call that popcorn lung. And basically it's sealing the lungs so you can't get oxygen, and that's almost worse than smoking. Studies are still out, but I just wanted to make sure you guys are aware of that. None of you are dumb enough to do that, so that's a good thing. All right, you ready? Was that exhilarating? Alex, was that absolutely exhilarating? Okay, here we go. Let's go backwards. Let's start at this slide. Okay, so the big question that you're all asking is, how do things move in and out of a membrane? How does oxygen move from that alveoli into my bloodstream? Okay, and the simple and easy answer is it just simply moves through the membrane, but it's a lot more complex than that. So we're going to make it 10 times as hard. Are you ready? Okay, so this is the special molecule we're talking about, and this is called a phospholipid. So I kind of write it right there for you. And a phospholipid has a phosphate head, which is a hydrophilic head. Phil means love. So if I'm anthrophilic, I love humans. If I'm hydrophilic, what do I love? What's hydro? Alex, what's hydro?
Water.
Water. So the top of this head, this guy loves water. Loves water. Okay, hydrophilic. This part of it is hydrophobic. And phobia is a fear, and then you have water. So this part of it is fear of water. So they call this a polar molecule because it's really two sides. It'd be like similar to saying, all right, let me find someone else to pick on. Who should we pick on? Let's pick on, who's fun to pick on? I'm not supposed to pick on students. It's kind of fun. Ashley, are you there? Ashley's not there.
Yeah.
Okay, Ashley, are you a Cubs fan or a Sox fan?
Cubs.
Okay, can you imagine if your body was cut in half and the top part of your body was a Cubs fan and the bottom part of your body was a Sox fan? Wouldn't that be bizarre?
Yeah.
Yep, it'd be weird. So that's what this molecule is. This molecule is part loves water and this part absolutely hates water. And water is a really big deal in human systems. So we're going to see how that functions here in a minute. Okay, so this is what's called a phospholipid bilayer. So here's your phospholipid. Here's that one head, and then there's that nonpolar tail. Okay. And basically what they are is this part loves water and this part hates it. So when you dump a couple thousand of these molecules in water, they arrange themselves. They literally arrange themselves. If we're in at a Cubs game, if we ever get to go to a Cubs game again, and we said, okay, and there's a whole bunch of people in one room, there's Sox fans and Cubs fans, what will naturally happen? What will the Cubs fans do? Who will they hang out by? Somebody answer me, so I'm not all by myself.
The Cubs fans.
Yeah, they're going to hang around other Cubs fans. And the Sox fans are going to hang around other Sox fans. And every now and again, they'll kind of give a little shout back and forth, but you get the idea. Well, these things do the same thing. They love to hang out with each other, but they like to be similar. So these non-polar tails, the ones that hate water, hang out with each other. And the only way they can do that is if they form this bilayer, um, this weird bilayer. Now, to make this work, this is really hard to do in your brain, but you got to try to do this. I want you to imagine a water balloon, okay? And inside the water balloon, I filled it with blue food colored dye. So you got a water balloon with food coloring that makes it blue on the inside. And I'm going to throw it in a swimming pool, okay? The blue will not move into the rest of the water. So it's always going to stay blue inside there because it can't get through the balloon membrane. When you think about this membrane, I want you to think of this part is like looking at a cross section of that balloon. Okay, so this, I think I'm going to run out of battery here. So this is the skin of the balloon. So this would be the inside of the balloon, and this would be the outside of the balloon. Okay. And what they're going to do, it's not just such a flat picture that I want you to see. I want you to see a more three-dimensional balloon picture, and this is the skin of it. Okay. Now, inside, like this is the membrane that allows the water not to move back and forth through it. This part here. That's the non-polar, the hydrophobic part. Doesn't let any water go through it naturally. Okay. So when we look at that as a membrane, it gets a little bit more complex. So here is our cell. Let's just kind of slow down here for a minute. Here's our cell. And here, see this little tiny box right there? That little tiny box is highlighting the cell membrane. And then it's showing you, okay, this is what that membrane actually looks like. So take a minute and just kind of slow everything down. And I want you to find a phosphate head in this. You find that phosphate head and then find that phosphate tail. So if let me change colors. I don't like the red. Go blue, that's pretty. All right, so this right here is your phosphate head. And then these are all your phosphate tails. So this part loves water. This part here hates water. Okay? So going back again, this part down here loves water. And inside your body, you are water. Okay, you're basically water. So inside the cell, this would actually be inside the cell here. That would be all water. Outside the cell would also be all water. But what a cell membrane does is it allows them to package things so that they could work more efficiently together. And we'll talk about that later. But the big picture right now is the membrane. Now you could see it's not as simple as it looks. Okay, it's more of a mosaic pattern. And if you've ever seen a mosaic pattern or a mosaic picture, it's where they take a whole bunch of little tiles and they make a picture out of it. In this case, you have a lot of different things all over the membrane. And this slide, you are responsible for on the test. So I will literally take this slide and I will erase parts of these words, and your job will be to fill in what they are. So where it says glycolipid, I will literally cross that out, put a box over it and say A, and then ask you what figure A is, like I've done on all my other tests. Okay, so you absolutely need to know this slide. Got it? Okay, so you notice these blue guys. These blue guys are proteins. And there's a whole bunch of different types of proteins. And these proteins basically do a lot of different jobs. This one you can see looks like it has a tube through it, so things can kind of move through. This one just is a big, it's a trans protein that goes all the way across, a transmembrane protein. It goes all the way across to both sides. And then you have others like this one that are just an inside surface protein, but they all do different jobs. And we're going to talk about those jobs in just a minute. Any questions on the cell membrane? So if I ask you a test question, Elena, can you see me winking? Are you able to see that? If I ask you a test question of what is the membrane composed of, you would tell me phospholipid bilayer. You would tell me different proteins, and you would tell me cholesterol. See the little yellow guys that are in there? Cholesterol. Cholesterol is in there to make it so it's not too rigid. And this is actually like it's similar to I don't know what's the I know where's Bingo? Bingo, where are you?
I'm right here.
Do you ever play with those things where it's like a whole bunch of little metal parts and you put them onto your face and it forms directly to your face? You ever do that?
I've never done that before, no.
But do you know what I'm talking about?
I think so, yeah.
That's exactly what a cell membrane does. It's very movable and it's very fluid. So if you kind of poked on this membrane with something, it would literally kind of move out of place and then just kind of come right back to it. So it's not a rigid thing. And it's not a rigid thing because of the cholesterol that's in there. You got it, Ava? Okay. I really wish I could show you this video, but it won't work. I'll get it to work later. All right. Let's skip that one for now. Let's do it. Okay, so let's talk about the different types of proteins. This is going to be a little tough, but I think you'll be okay. This is also on your test, so you definitely need to pay attention and write these down and write down examples. So when I'm talking about proteins, the different protein types I'm going to list here. So when we went back to that picture, that's those blue little structures that are in the phospholipid bilayer. So these proteins basically are doing all of the work. Okay, they do a lot of different work for the cell. One of the things they do is they act like a channel. And a channel is just to allow particular molecules to go in and out. Okay. The example I'm giving you is a chloride channel. So chlorine is a really, really important chemical in your body. It helps you sweat. It helps you move mucus out of your lungs. It does a lot of important things. And it has to move back and forth in and out of the cell all the time. People learned about this because they found out if it's broken, you get a disease called cystic fibrosis, which is a very challenging disease, very tough disease to live with, but definitely you can do it. And cystic fibrosis is basically when that chloride channel doesn't work and you can't move chloride back and forth across the cell membrane. So you have incredibly salty tasting sweat on your skin all the time. And you also have really bad mucus built up in your lungs. It's very hard to get rid of. Really hard to get rid of. Did you guys see that movie? I think it's called 5 Feet Apart. I think, I'm not 100% sure, but I'm pretty sure those two kids had cystic fibrosis. Am I wrong or am I right? Anybody remember? I think they did. I don't know. But anyway, it's a very tough disease to live with, and it's basically due to the chloride channel. So let's go to the next one. Carrier protein. And some of these are labeled really well. So like channel means things can move back and forth through the channel. And it's always specific. It's never just like an open road. It's more like a border where people are letting you come or go based on criteria, such as is it chloride or is it not chloride? Like if it was fluoride, it wouldn't be able to move through. Well, carrier protein does that same thing, but instead of using it as a channel, they grab it and they literally spin it into the cell. So it's a mechanism where they actually like grab it, kind of like a what's a good way to think about this? Just a mechanical arm that's going to grab a particular protein or sometimes glucose. And so literally going to grab it and then kind of turn it inside the membrane and drop it inside the cell. Sometimes they grab it from the inside of the cell and they want to move it out. Okay, and we'll talk more about that later. But a carrier protein interacts with very specific molecules to cross back and forth. And we'll show you examples of that in a minute. All right, this is a tough one. Cell recognition protein. Cell recognition proteins are basically on your membranes. And let's see, who haven't I picked on? Seems like I pick on the same people. Let's pick on someone new. Who haven't I picked? Bella. All right, let's say Bella was driving down the street and a helicopter crashed into her car, and you lost her liver. Okay, Bella, you okay with that? Okay, so Bella lost her liver, and we need to get her a new liver. So if they just decided to take someone's liver and just put it in Bella's body, all of the cells in Bella's body, her immune system would attack that liver and destroy it because it's foreign. So the question then becomes, as a scientist, what makes it foreign? What makes something in our body foreign? And the reason why our bodies attack foreign things is they literally will search them. They'll look at the plasma membrane and they'll try to read it like a scanner. If they scan a particular protein that says, yes, I am Bella, then they won't attack it. If they scan it and it says, oh, hey, Brody's going to give her the liver, and Brody gives him part of his liver to her, immediately Bella's like, no. And her system will attack it. It'll read Brody's liver cells and say no, and it'll destroy it and ruin it. That's why a lot of people, when they have to have an organ transplant, they usually go to someone closely related to them, or they try to find someone that has a match. And when they say a match, they're talking about the proteins that are recognizing the immune system on the cells. Okay. So it's cells with foreign, they call them major histone compatibility complex, but you don't need to know that. Just MHC is okay. But it basically provides for our immunity. Sometimes if people have organ transplants, what they have to do is they have to dim down the immune system so it's not so active, because nobody's a perfect match usually. They usually have to dim it down a little bit so the immune system's not so attack mode all the time when they do an organ transplant. So Bella, how would you summarize what a recognition protein is?
It's like a protective one that like protects you from foreign stuff.
Yeah, I think so. Do you remember who's an athlete here? Raise your hand if you're an athlete. If you're an athlete and you go to that first day of practice and everybody has their really fancy water bottles, you know what I'm talking about? And your parents put your initials on it, like if it's Carter, Carter, what sport do you play?
Baseball and hockey.
Okay, so he's going to baseball practice and he's got his baseball glove or his water bottle. He sits there, and his mom puts his initials on it, CK, and that's so nobody else mess with his stuff, and they know it's not theirs, it's his. That's basically what your cell is doing too. It's basically putting their initials or their name on the cell, so it's always recognizable. Good job. All right, receptor proteins. Receptor proteins. Well, let's talk about human growth hormone for a minute. How many of you, raise your hand if you've heard of human growth hormone? Human growth hormone is released by our pituitary gland, and it goes into our bloodstream, and it basically goes to all our cells and says, okay, during the growing period of your life, I need you to get the cells to get bigger, get longer, and reproduce more quickly. At my age, I'm not growing anymore, so I don't have as much human growth hormone released. But it's basically a growth hormone. What a receptor protein does is it accepts a very specific message, such as human growth hormone, and it basically accepts it, and it sends information to the cell to do the job. So for example, let's say Carter is playing hockey and he wants to be 50 pounds heavier and 3 inches taller. So he takes some human growth hormone. That hormone would go through his body and it would basically go to every single one of his cells, and it would go to the human growth hormone receptor protein on his cells. And then those would send a message to a cell saying, okay, continue to grow and continue to divide and get bigger and bigger and bigger and bigger. And people do this. People literally do it. Sometimes it's by doctor order. Sometimes athletes take it illegally. Carter, don't ever do that. It's bad for you. But it is definitely something that some people do. Barry Bonds was famous for doing this. Barry Bonds took human growth hormone and his legs and his calves got really strong and big, but his knees did not. So he has incredibly weak knees right now. He went up, I think it was a size and a half in helmet size, because even his head got bigger. Everything was growing, and it doesn't do it proportionately, and that causes a lot of problems. So human growth hormone, the idea is that a human growth hormone receptor basically receives the message. An example, yeah, I'm going to run out of battery. An example in my case is I'm a diabetic and I have to take insulin. Well, my body has insulin receptors on the cells that basically take in insulin and they receive it. So a receptor protein is basically just receiving messages from your body. Last one, enzymatic. Enzymatic are proteins that basically catalyze a reaction. I would write down an example of this is your ATP synthase. Remember how we had that in the membrane before? And it basically took the message and it basically took ADP and turned it into ATP. That is an enzyme. It makes something or it breaks something down. Got it? Shake your head vigorously. Yes, if you get it. Study the slide. It's an important slide. All right. So both these scientists are 6 feet long. 6 feet tall, pardon me. 6 feet tall. Now, one is in a tribe that has very similar height patterns to everybody in the tribe. And he's 6 foot tall, and these people look like they're maybe 4 feet tall. What could you tell me about their human growth hormone receptors? Do they have a lot of them or very few of them, and why? Awkward internet wait time is happening.
They have a lot.
These guys, do you think these guys have a lot of human growth hormone receptors? Do they look like they're growing a lot?
Oh no.
No. So I think it'd be the opposite, right? They're going to have very few of them. And because they're in a tribe, and it's a very isolated tribe, everyone has that same kind of level of human growth hormone receptors. Here's another tribe in South America, and basically everyone's this tall. So this guy is 6 foot tall. I don't know, how tall do you think this guy is? Maybe 7.5 feet tall? And everyone's that tall in that tribe. And the reason why is because they have more human growth hormone receptors. Okay, so human growth hormone receptors is a hereditary trait. So if you have, if your dad is 6 foot tall and your mom's rather tall, there's a good chance that those receptors are going to go to your body and you're going to be similar in height too. Okay, so that's just kind of how that receptor part works. I just thought this was kind of a cool example. All right, we're almost done. You tired yet, Emerson? Is this a lot of work? Okay. All right. So, cell membranes, what can go in and out? I would write this down on this slide. This is another very important slide. I could summarize this slide very simply. Things that can move into and out of a membrane are small and or uncharged. I would write that down. Things that can move into and out of a membrane are small and or uncharged. Okay, so for an example, water is a small molecule. Water can move back and forth across the membrane freely. Let's say this is oxygen. Oxygen is uncharged. It's O2. It doesn't have a plus or a negative. It cannot, pardon me, it can move easily back and forth across the membrane. CO2 is small. CO2 could also move back and forth across the membrane. Now, if I have a large molecule like glucose, let's say this guy's glucose here, glucose cannot move through. It gets repelled by the membrane. It's too large. So to get that into the membrane, you would need either a channel protein or you would need a carrier protein to get that in. Something that is charged has a positive or a negative. Chloride, I don't know what that happened. You guys see that? I don't know what that meant. Chloride is negatively charged, so it cannot get through the membrane. So all of a sudden now this thing's starting to say what I say. You see that? Did I do that? That was pretty cool if I did that. Let's say I did that. Okay, so captions for those of you that need captions, I've just provided for you. If it's charged, it cannot move through the membrane, so it's going to need a channel. So remember we said that like if we had a channel protein kind of look like this and it had a hole in it, that would be for cystic fibrosis. So let's say it's chloride could kind of move through that without going through the membrane. Okay. So the best slide we have for that, let's go to this slide because we're almost out of time. We'll talk about this and we're going to talk about how I'm going to turn this into part of your test. How much time do we have? Okay, we have 10 minutes. All right. So here is simple diffusion. And simple diffusion means I can go from a high concentration to a low concentration on the other side, and I could literally go right through the membrane. Okay, that's simple diffusion. That would be oxygen, that would be water, that would be anything that's small and or uncharged. Okay, if it's something's large, like these cases, these guys are large, this is carrier mediated, which means helped, facilitated diffusion. So here you have a large molecule going into this side, and then it gets carried and pushed out onto the other side. But it's still diffusion because there's a higher concentration here than there is here. The next one is channel mediated. So again, channel mediated facilitated diffusion. Facilitated means helps. So here's our channel. This would be like a chloride. Let's say that this had a negative charge to it. Chloride could literally move through there from a high concentration to low, but it couldn't go through here. It would get repelled here, but it can move through the channel. So this one's a channel. And then our last one is the very special rule of osmosis, where water can move back and forth across the membrane or go through what's called an aquaporin, which we'll talk about later. Aquaporins are in your intestines. I think we talked about diarrhea, right? That's basically what's in the intestines to kind of increase water into your in your intestines if you need it. Okay, so just so you know, I could take this and I could turn this into a test. I could easily take each of these pictures and mix them up, and I could have you match up the picture with simple diffusion, carrier mediated, facilitated diffusion, channel mediated. So I'm going to have you take this part of the test and match up. So I put a star on this slide, or star saying you need to know the passive transport slide, and I think you'll be in good shape. Any questions on that? This is, I think, 4 questions on your test. So make sure you know it. Okay, this is going to be the last slide we do, and then I'll leave you guys alone. And believe me, we're going to come back through this and go over it again. Yeah, we got 8 minutes. Okay. Passive transport. Passive transport is the movement of particles without the use of energy. If a student, it means you don't like to put any energy in the class. And that happens. Passive transport is you do not use ATP energy. Everything is just naturally going to happen. The best example I can think of this, to understand it from a molecular point of view, is diffusion. For example, imagine that I have a huge garbage bag full of ping pong balls that have a motor in them. So every single ball is constantly vibrating and moving. Okay. And I'm sticking them inside a big, huge black plastic bag. Okay. So what I'm going to do now, remember, the ping pong balls are constantly moving, always moving. They cannot stop. But when they're in that plastic bag, they're moving really fast and they're just bumping into each other really fast and not going anywhere. If I open the bag in a second, Brady, what would you imagine because I'm in my dining room right now, what do you imagine it would look like if I open the bag all of a sudden? In 1 second, I took a picture, what would I see? Would the ping pong balls be close to each other or far apart?
Close in this case?
What'd you say?
Close?
Yeah, they'd be incredibly close. And if they're close, if they're always moving, they're always going to run into each other, right? They're going to have a lot of collisions, tons of collisions. Now, if you come back in a half hour and I close the door to my dining room and you took a picture of the room and these things can kind of float and move, what would you see? Would
They'd
they
be spread
be,
out far apart.
yeah, they'd be evenly spaced out through my room. Would the ping pong balls still be moving?
Yes.
Yep. And they're constantly going to be moving, bumping into each other. Now, diffusion is the process of the net random movement of molecules from a high concentration to a low. So when Brady first took that picture, those ping pong balls were in a very high concentration, all clumped together. He came back an hour later and they were all spread out. So what they had to do is they diffused, right? Those ping pong balls went from a really high concentration to a really low concentration. And they did it naturally. They just kind of bumped into each other and naturally moved around. Okay. Osmosis is this same movement, but when it happens with water through a membrane. And we're going to talk about that in a little bit. And then finally, facilitated diffusion is when it gets help of a protein. It could be a carrier protein like we talked about, or it could be a channel protein, which we talked about. Okay, so facilitated diffusion is diffusion, but now we're using a protein to kind of help it along. And you need a protein if it was a large and or charged particle. Whew, that's a lot of stuff. You guys exhausted? All right. So, any questions on this? It's a lot of stuff.
I have a question.
Good.
So, like with the cell recognition proteins, with like a lot of autoimmune diseases, does that mean that they just like didn't work and they like attack the part of
Yeah.
the
Well, that's what type 1 diabetes is. And that's what I have. So, there's a certain cells in the pancreas that make insulin. So, an autoimmune disease basically means my immune system did not recognize those cells in my pancreas as me, and they literally attacked and killed my own part of the pancreas and destroyed the parts that make insulin. Other autoimmune diseases, such as trying to think of a good example. Anyway, any autoimmune disease can literally turn your immune system against you and ruin part of your body? It's a great question. Any other questions? I can't remember the disease on that. So in the next couple days, I'm going to let you take the quiz. You need to get your vocab done. At this point, guys, you cannot miss an assignment. You absolutely have to be on your A game. You absolutely have to get all your work in. Okay, it's critical. We're almost done. We got less than 2 weeks. A game stuff right now. I'm not going to make it hard on you. I'm not giving you a final. But this unit, don't miss a thing. You got it? You cannot let this grade slip. You guys are too good of students to let that kind of stuff happen. All right. So without anything else, I know that was a lot. We'll go through it with a worksheet to practice it. It's going to be an open note test, so don't totally freak out. And we'll go from there. You can sign out and wave goodbye. Bingo, put your lungs back in.
All right, I almost forgot to.